Okay, recording has started. Now I'll share my screen. Okay, can everybody see this? Or some people?
Yes, looks good.
Okay. Yeah, so I kind of changed the title to make it even more provocative, and so people can kind of tell me that I made the wrong title later too. So oxidation of long chain fatty acids dispensable for survival of the retina and RPE? Question mark. It's not an answered question, but it's something that we're trying to gain insight into with the data I'll present. And I have some names listed here. It's not actually comprehensive because this project has existed sort of well before I knew about it, and I kind of came in and helped. And so really, it's kind of a highly collaborative project that sort of, I think, originated with a lot of work that Nancy did. And, obviously she's not the only one. And it's sort of based on, you'll see what it's based on. But I'll just advance the slide. There we go. So yeah, I mean, you can tell from the title, obviously, and it's about fatty acids. They're considered a source of energy, a component of lipids, and specifically interested in long chain fatty acids for this talk, which, I'm kind of saying is 13 to 21 carbons long, although I don't know sort of the exact cutoff. And they're delivered to the eye in a couple different ways via circulation. You can get them through lipoproteins, through fatty acids conjugated to albumin. I don't know if exosomes substantially contribute, probably not substantially, but perhaps they do contribute fatty acids through their lipids to the eye. And if you're thinking about the RPE, it gets a dose of fatty acids from photoreceptors in the form of outer segments. And I kind of looked at a few papers to get an estimate of, what proportion of outer segment fatty acids would be long chain. And there are different estimates, but the sort of average would be something around 14% is palmitate, and then, close to 19% is stearate. And, I don't need to be exact for that, just to say that, a fair proportion of outer segment lipids have fatty acids that are long chain. And so, there's this sort of burning question that I have that I'm not going to solve with this talk at all, but is there something that relates fatty acid metabolism to drusen, or a research topic I'm interested in also is, even if there isn't a direct relationship normally, is there something we can do to change fatty acid metabolism to alter drusen? But overall, fatty acids, they get in. What happens, they can sort of be exported, they can be incorporated into lipids, they can also be oxidized. And for long chain fatty acids, the main site of oxidation is in mitochondria. And so, to be oxidized in mitochondria, they need to be conjugated to carnitine by this enzyme carnitine palmitoyl transferase. And then they can enter in, and they go through beta oxidation to basically make, these units, these 2-carbon units of acetyl-CoA, which can be further incorporated into metabolic pathways, can be incorporated into the TCA cycle to make citrate, and sort of downstream in ketogenesis, it can make beta hydroxybutyrate. So that's kind of just what is the route for fatty acids when oxidized. And there's some evidence that they certainly can be oxidized in the retina and in the RPE. The oldest paper I could find is this Keene and, let's just say this Keene paper from 1965. And they used radioactive 14C palmitate. They provided it to ex vivo rat retinas. And they saw that, they looked at basically the different fractions that this 14C palmitate went into. And they didn't recover all of their sort of radiation that was imported into the retina. But they were able to show that, of the radiation they did recover, a substantial amount of the palmitate ended up as sort of esterified as part of a lipid. This is, again, isolated retina. There's no RPE present. And so you can see sort of, let me just enable the laser pointer for you. Okay. So this is kind of the average of all these experiments. And if we, set the palmitate uptake to 100%, then basically almost 40% of that radioactivity could be considered to be in fat esters in the retina. So the retina does take fatty acids and sterify them to make lipids, and 12.5% stays as a non-esterified fatty acid, and then they recovered maybe 10% of uptake in palmitate as radioactivity and CO2. So that's strong evidence that the retina can oxidize fatty acids. And then there's this other paper, this Tiny et al. In pediatric research, and they have several bits of evidence that suggest that the eye can perform fatty acid oxidation as well. Even especially long chain fatty acid oxidation. A figure I'm not showing shows that they used 13C palmitate, and they saw the appearance of 13C acylcarnitines from that palmitate, and that was in RPE, porcine RPE, I believe. And then they also have these human retina sections, which they sort of bleach the pigment in with hydrogen peroxide. And they show that in these retina sections, they have staining for the mitochondrial trifunctional proteins. That's not the nuclei. The nuclei are stained H&E, but you can see in the inner segment here, there's reasonably strong staining for the trifunctional protein. It's not present in their secondary only control. And in the RPE, which again was bleached, they see substantial labeling for the trifunctional protein that is not in the secondary only control as well. So there's this sense that, the retina is certainly capable of oxidizing fatty acids, and long chain fatty acids in particular, and that the enzymatic components that are necessary for mitochondria to oxidize long chain fatty acids exist in both the retina and the RPE. And more recent work that I bet all of you are familiar with from the Philp lab and from the Bose Battaglia lab shows sort of dug deeper into this, what's going on. So in this Adijanto paper, I have these two panels, and the one shows that if you provide labeled palmitate to RPE cells, human fetal RPE, they'll make beta-hydroxybutyrate. And they'll do it regardless of whether there's glucose there, but glucose seems to accelerate the formation of beta-hydroxybutyrate. And that's labeled from the labeled palmitate. And then in this middle panel, you have retinas fed with labeled beta-hydroxybutyrate, and you can see that beta-hydroxybutyrate is incorporated into metabolism in the retina. So you have this sort of sense that the RPE fatty acid oxidation could generate this thing that is then used by the retina. And then this paper from the Bose Battaglia lab took it a step further and showed that outer segments alone, or in addition to glucose here, but outer segments are able to generate beta-hydroxybutyrate as well. So there's this idea now that if you have human fetal RPE or RPE in general that's fed with outer segments that contain all these lipids and fatty acids that are on there, including long chain fatty acids, that they can oxidize those fatty acids, make beta-hydroxybutyrate, and that beta-hydroxybutyrate can then feed the retina. That's sort of the general idea. Okay, so we then I say we, but I entered into this project late, so this is something that I think that Nancy really spearheaded. And the question was, these pathways certainly can exist, but what impact do they have on the overall sort of health and function of the retina? That was the question that got asked next. So, and that's a question that was asked both of whether or not ketogenesis is essential for sustaining the retina, or RPE ketogenesis in particular, and fatty acid oxidation either in the retina or the RPE. And so, I think I've showed this figure to some of you before, but, we basically have a system where we can measure fatty acid oxidation to some extent by, taking labeled a retina or an RPE choroid in this case, putting them in a dish with labeled palmitate, and look at the products of that labeled palmitate either in the medium and see the accumulation of that over time, or in the tissue itself. And the labeling from palmitate can be incorporated. Again, it can make acetyl-CoA and make beta-hydroxybutyrate, which will either be labeled sort of two of those carbons will be labeled by the two carbons of acetyl-CoA, or they'll mix together with unlabeled acetyl-CoA and make, BHB can be labeled either at 2 carbons or 4 carbons, depending on which acetyl-CoA it uses, the labeled form or the unlabeled form. And the same is true of citrate. And I won't dwell too much on that, but basically we use it as a way to look at, how well palmitate is utilized. And, this is just sort of a proof of concept. We provide more labeled palmitate in this sort of middle panel. That's just a different concentration here. We see a greater proportion of beta hydroxybutyrate is labeled. Here, M+2. So again, 2 of those carbons, acetyl-CoA carbons that made BHB were labeled. And we can shut it down with the inhibitor of the, sorry, mouth's getting dry. We can shut down labeling of BHB when we use etamox here, an inhibitor of carnitine acyl transferases. Okay, so pretty simple experiment. This was in RPE choroid, the mouse. And so, we're using this system and looking at this system and other systems to look at whether or not RPE ketogenesis is essential for sustaining health of, in this case, the retina. And so, again, this is kind of just the pathway by which acetyl-CoA, this is from Wikipedia, but pathway by which acetyl-CoA can be used to make beta hydroxybutyrate. There are a lot of steps here, and the one that I want you to pay attention to is this HMG-CoA synthase step, which transforms acetyl-CoA and acetyl-CoA to make HMG-CoA. That is a rate-limiting enzyme in ketogenesis, and it's something that Nancy knocked out, essentially. So she had these mice with floxed HMG-CS2 and an RPE65 pre, and I think I have ERT2, and I'm not sure that's appropriate. But regardless, these are RPE cells that when the Crete is active, there should not be any HMG-CS2. So, here I have a Western blot from our lab showing that, you can sort of use this RPE delta HMG-CS2, and there's still there's no HMG-CS2 in the retina to start with, so it has no effect. The RPE has HMG-CS2, but when you use this transgenic model, it doesn't have it any longer. And, it seems like, relatively speaking, the GLUT1, MCT3 are sort of maintained. And so, this is just to show that the knockout kind of works. And you can also see the confirmation in this immunofluorescent image, where the labeling is for HMG-CS2, both in floxed controls and in RPE delta HMG-CS2 RPE. So, okay, the knockout works. Great. What metabolic consequences does that have? Theoretically, it should really stop the ability of the RPE to make ketone bodies from fatty acids. So we're using this labeling strategy that I showed you and looking at the incorporation of 13C from palmitate into intermediates in the TCA cycle, as well as ketone bodies. And what you'll notice is that if you look at tissue metabolites, really there's not a huge change, not a huge effect of this knockout on even labeling of BHB. And so I think the instinct was first to think that the knockout didn't even work. We looked at BHB, though, that was exported into the medium, and we did see, so we looked at a few things exported in the medium. Among them, in this sort of lower right-hand corner, is BHB. Again, HMG-CS2 is sort of necessary and rate-limiting to make BHB. And there is a reduction in the formation of BHB over time, but it's, not totally shut off. So that indicates to us that, the knockout does work, but it doesn't seem to be stopping all BHB production in mouse RPE choroid. And, I think there are two hypotheses that we really need to test. The first is that, there are other cells essentially making BHB in this preparation, which wouldn't surprise me whatsoever. We're pretty careful to cut off things like muscle and connective tissue when we're doing these experiments. Theoretically, they could contribute, but it could still be other resident cells, choroidal endothelium or something like that. And then the second idea is that perhaps BHB can be synthesized by an alternate route. And, I have little maps of ways that it could potentially be working, but it seems most likely that we should test this idea of other cells making BHB first. And, but it was kind of okay that other cells were making BHB in this preparation, because the RPE cells are the things that they're sort of seal the retina off from the rest of circulation and all these other cells. And so my thinking was, if there's still an intrinsic deficit in BHB production by RPE cells, then it should still affect the retina, because it's sort of a different compartment that interacts with the retina. And so we did look at, I'm using the royal we here. I didn't look at these, but we looked at the OCTs from mice with their control mice or without HMGCS2, and this one is 3 weeks post tamoxifen injection, but we have OCTs up to about 5 months of age, and we see that there isn't really any meaningful change in the thickness of the retina or thickness of individual components of the retina that we can quantify. So, I think there might be an eensy-weensy bit of change by the latest time point, but we kind of have to do a much more thorough job to be certain of that. Regardless, yeah.
Sorry to interrupt, but is there a total body knockout of this gene in mice?
There might be. I don't know.
Just be interesting if there's a phenotype of a full body knockout, if it doesn't, loss of it doesn't seem to affect production of your beta hydroxybutyrate very much.
Yeah, I mean, that would certainly tell us whether or not there's an alternate pathway for beta hydroxybutyrate production. And that would be a neat thing to test. But I am unaware of this model if it exists. And that would be good. Yeah.
Dan, there is someone in Australia who made a full body knockout that years ago, 10 years ago, we were going to get it from her 12 years ago. And then, getting animals from Australia at that time was difficult. And there was just a paper that came out on zebrafish retina, and I'm just blanking on exactly what they found, but there was a phenotype in knocking out HMGCS2 in the zebrafish retina.
Okay, good to know.
Dan, do you think that in 3 weeks, you will really see a retina degeneration? Because 3 weeks seems to be short.
Yeah. This says 3 weeks, but it's been tested up to 5 months, and the retinas essentially look the same.
Okay.
Dan?
Yeah.
Can you hear me? Just on the previous slide, the what do you think is going on with the glutamate?
Oh, dude, I have a bunch of pathways drawn and ideas sketched out for how that could work, but it looks kind of like what you imagine a madman would, sketch in, I don't know, crayon all over a wall or something. I certainly,
Dan, I take that
sure.
personally.
So yeah, I think there's an idea of what could happen, but it's not tested, and it's pretty complicated. So I showed you the data knowing that maybe it would come up, but we certainly don't have an answer for sure.
Okay.
Dan, have you looked at the RP3 mice itself? Just the Cree itself, if it will affect the OCT or ERG?
This is something that John would know. John, are you here?
He's
I know
on.
there are some Cree models that certainly is a problem.
That has been done when the RPE65 Cree was established with the ERGs, and we've done OCTs on Cree mice treated with tamoxifen and haven't seen changes.
Okay, so it's not the Cree.
Well, again, we're not seeing any intrinsic change yet that we could even attribute to the Cree. But we will later on see something that I'll show you.
Yeah. So, Have you tried any like stress challenged condition instead of just looking at the normal development?
Yeah, I mean, I guess that should have been in future directions, but it's not. But that's totally. So I think both overfeeding and starvation are conditions that it would be worthwhile to test. I think I'm most interested in starvation because that's when the body theoretically, shifts to using ketones as a fuel. But there is a sense that, the body likes to preserve glucose for neural tissue. And so you might have to take it out quite a while. And so I'm not sure. I think it would be worthwhile for sure, but we haven't done it yet.
Yeah, so in our studies, well, we look at like different hormones and also enzymes involved in this lipid metabolism. What we notice that like in the normal development, so we do like in the first 1 or 2 months, looking at the neurons and the vascular changes, they are pretty happy mostly. But if you're challenged with, so in our model is oxygen challenge and the diabetic challenge, so you will see a difference. So you can see the metabolic adaptation was affected. Yeah.
Yeah, I mean, I think it's it's reasonable to say that, even if there isn't a change normally, that it could be a sort of a stress response pathway.
Yeah.
But the question, at least for now, is it even useful normally? So I think we have to kind of inch our way over to stress responses, but first have a full characterization of
Yeah,
what happens
that's for sure.
physiologically.
Yeah.
Dan, sorry. Have you looked at the mitochondrial morphology or other markers for the RPE to look how they look in these mice?
So I think the markers for the RPE by Western blot, some markers look reasonably good or unchanged.
Well, it looks like you have less beta-actin in your knockout. So it could be that your MC3 is much lower, your GLUT1 is also lower. No? Or is it just
Well, if there was less actin, it would sort of mean that, I mean, I think there is a little bit less MCT3 and a little bit less actin, and they sort of cancel each other out. This has been quantified, and I didn't show the quantification just so I didn't have too packed of a page, but I don't think MCT3 has changed all that much. I think the main concern I'd have here is retina GLUT1, but I don't really know what the actin would be like if it was sort of, we boosted up that signal a little bit. So I don't want to make a comment on it. But I think, this change in actin probably isn't substantial enough to, I don't know, to totally erase a band if it was present. There is a small band here if you kind of squint. And I've validated this in other samples as well, that there is still in the RPE chloride some HMG-CSF2, but that's not really your question.
No, I was thinking about other markers, other transporters that could, and it looks like maybe they are a little bit downregulated. So
I think relative to actin, they're probably pretty similar, but it's kind of tough to say.
okay.
But you really have to do a more comprehensive experiment to get a sense of all the transporters, and so we haven't done that. So it could certainly be that some things are altered.
Dan, along the same line, if you go to the previous page, I mean, your Cree looks really very nice, that you have quite significant knockout, but then when you are looking at the beta hydroxy, you are using the RPE choroid sclera, so your hypothesis that it could be coming from other cell type is true, potentially true. Is it possible to isolate the pure RPE cells from those knockouts and do it without a choroid sclera contribution?
The only way that I can think to isolate the RPE cells would be some kind of enzymatic treatment. And the concern is always that the enzymatic treatment is going to sort of change what the cells are intrinsically doing, because you're essentially putting them in a situation that might be different from their sort of physiological one. We know that isolated RPE cells certainly do use palmitate and make beta hydroxybutyrate. But I guess you're asking the question as to whether or not the ICUP without RPE cells is doing the same.
Right.
I don't have that data.
Okay.
Hey, Dan.
Yeah.
I have a long detour, which can get you to that answer eventually.
I'm sorry, I can't hear you.
Can you hear me now?
Yeah, I can hear you, just maybe a
Okay,
little.
there is a detour to actually generate an in vitro model. You can essentially generate iPSCs from your RP, CRE, homozygous LOXP mice, either using blood or fibroblasts, and then you make iPSC-derived RPEs. So that can drive CRE recombinase in a tamoxifen and RP65 dependent manner. So then in your in vitro system, you can just do a short-term tamoxifen treatment of your cells, and you have a pure in vitro model.
Yeah, I mean, I agree. I don't think we need an in vitro model of RPE. I think what I would love is an in vitro model or some other model of the sort of choroid, sclera, all these other cell types that might be contributing to a response. But it's certainly, you know, I agree, that's certainly a feasible experiment.
The other thing I'm thinking about is, so you have knocked out HMGCS2 in the RPE, but so you have essentially completely shut down the mevalonate pathway. But the
what
RPE,
pathway?
the mevalonate pathway, which feeds into cholesterol.
No, HMGCS1, I think, is what mediates that pathway. No?
I'm unsure. I don't know what the cholesterol synthesis fate is going to be in your HMGCS2 knockout, but it's not going to, the effects, whatever pathological effect you may or may not observe in this model, it's not going to come because of lack of cholesterol synthesis, because the RP is happily taking up lipoproteins.
You're saying in this model, the RPE
So
is...
in this model, cholesterol synthesis in the RP, cholesterol homeostasis in the RP may not be affected because it's also importing lipoproteins.
Yeah, I mean, it's almost a good thing, right? Because we want to narrow in specifically on ketone synthesis. And I do have a sense that perhaps the cytosolic cholesterol pathways would be maintained because I think they use a different form of the enzyme. But yeah, let's move on because I have even more things to sort of argue over, which not to say that any of you are trying to argue. You're not. But I kind of want to argue about some of these other things. So okay. So again, you know, I don't think this is a sort of a conclusion yet, but we'll just frame the question as if ketogenesis isn't essential for eye function, and you know, we really need to go further to make sure that that is true. Is beta oxidation essential at all for the eye function? Is mitochondrial beta oxidation specifically? So that oxidation of long chain fatty acids. And so this is also using the RPE65 line, CRE line, and knocking out, instead of HMGCS2, CPT1A, which is the enzyme that's rate limiting for long chain fatty acid oxidation. So you need it to conjugate the palmitate or any long chain fatty acid to carnitine. And in this experiment, you know, we have just imaging of, I think this is RPE choroid, mouse RPE choroid that has been given the CRE, or, you know, in the CRE model or not the CRE model, and you can see CRE localized into the nucleus. And I assume this must be tamoxifen treated. But regardless, you can see in this RPE delta CPT1A knockout that there is, again, a reduction, but not a total wipeout of CPT1A. And so it's the same sequence of experiments as before, essentially, where we're looking at metabolism in the RPE choroid, and we're doing it with 13C palmitate. And here you have what effects that look much more substantial. So there's less beta hydroxybutyrate that's made in the tissue. There's less citrate. Not everything has changed, again, because I think that the formation of some 13C intermediates is happening in other cells. And you can see that the flux of 13C on things into the medium is also reduced, but not absent. So it's the same story as with this HMG-CH2 model that you just saw. There is a decrease in utilization of palmitate to make, beta hydroxybutyrate, as well as other things in the TCA cycle this time, whereas the TCA cycle flux wasn't impaired in the HMG-CH2 model. And I also looked at glucose and lactate and quantified their uptake or output, and that wasn't any different. But the whole idea is that you can wipe out CPT1A and you have metabolic effects that essentially you would have predicted, because there is still some CPT1A in the tissue, in these other cells that we need to study, and they might be doing some proportion of this, but a lot of metabolism of long-chain fatty acids is decreased. And so the same question was asked. This is in 8-month-old mice, and I think they were transduced with tamoxifen at a young age, although I don't remember off the top of my head what that age was. And basically, we're not seeing any difference in retina thickness by OCT or autofluorescence. We don't see any real change, perhaps a slight reduction in fluorescence intensity, but nothing sort of substantial that would indicate a pathology when you remove CPT1A from the RPE cell. So that's kind of the story for the RPE, that you can get rid of in those cells either CPT1A or HMG-CH2, and it has a metabolic consequence, but it doesn't seem to cause retinal degeneration. And so I think the question that Kira asked is particularly it follows really well from this data, right? So they're not essential normally, but it might be that these pathways become essential when there's some sort of stress.
Dan, would you prefer I not ask a question, or?
Oh, go for it.
Okay. I'm embarrassed to not know, with talking to you and Nancy this much. Did on the HMG-CH2 knockouts, does the rate of palmitate consumption in the eye cups, the depletion rate, change? You showed that there's less of the beta hydroxybutyrate being produced, and so I presume that means it's also being consumed less. But do we have the corollary of measuring the depletion rates of the palmitate in the media?
I don't think it's any different, but I have to look back at the data to be sure.
I guess it could be not different because still only a small fraction is shunted to beta oxidation, and the rest is incorporated, and all the other things that can go with palmitate, I guess.
That's a strong possibility, but I don't think I got good enough, high enough levels of consumption to have a great visual of that I really trust.
Dan, I would say that's correct, and also the size of the pool, the difference that you would be able to perceive in what to utilize versus a steady state pool. I think it's going to be a small difference, so I don't think it's going to be statistically significant. In other words, you're following a tracer with a C13 label, right? So I don't, per Jason's comment, I just think you wouldn't be able to see that consumption rate.
Well, he'll be able to see the consumption of the C13 itself, but not if he's just following the C13 palmitate label.
But just to be clear, Jason, you're talking about from the culture, from the medium, essentially, that I'm culturing decoyed in, as opposed to sort of within the tissue.
The media pool is far greater than what you would be able to see. You can measure it, but I would say that it's going to be a small difference.
Yeah.
And also, Dan, do you have ERG data in addition to the OCT and like the founders data?
I don't think we have it for this model yet, although that is the long-term strategy.
Yeah, because when we look at we look at the lipid pathway, So it's very interesting. By 1 and 2 months old, you can see a decrease in ERG, but the thickness will change until probably 8 or 10 months old.
Mhm.
Yeah.
So wait until the next slide and you'll see some sort of preliminary data there. Or
Okay.
not the next slide, but in a couple slides.
And
Okay.
with your Western blot that you showed us just previously for CPT1, it looked like there was a significant amount of residual. Yeah, right
Yeah.
there.
Yeah, I mean, so first of all, not my Western. Again, I'm presenting like a lot of other people's data, and I hate this idea that I'm kind of automatically passing it off as my own. It's just language that I'm used to using. But again, the vast majority of this work was other people. I did the metabolism stuff essentially. And but yeah, this Western, there is residual CPT, and I imagine that that is the thing that is sort of causing the residual palmitate oxidation that we still see in the RPE choroid. And so you could, I think the overlap is very strong between the sort of Cree and the DAPI here, although I didn't have the overlap image to share. But so it suggests that the knockout is probably not necessarily, you don't see that necessarily because it's mosaic, but I think because other cells in this preparation are probably using palmitate.
Yeah, and I think the last thing that I was thinking about to address the possible multiple cellular sources that you have in this complex, because they're not pure RPE cells, is to do in situ hybridization and show that the signal, where is the signal and where is it being depleted from
Yeah.
on Cree.
Yeah, I think also just a simple, I don't know how good the antibodies are, but a simple immunohistochemistry experiment in sections from these mice would go a long way.
Right. We've had to address this kind of problem in some projects we've done where either there are no commercially available antibodies or they are unreliable.
Yeah.
But in situ works every time.
Okay.
Yeah. Thanks.
Thank you. Okay, so if this was kind of like, I don't believe you, then if anyone was thinking that, then just wait until this next one. So this is again yet another model where instead of knocking out CPT1A in just the RPE, it was knocked out in photoreceptors, or in rod photoreceptors specifically. So this is, again, sort of very similar models that we've been using. This is an ICRE that should be rod specific. You have a much more drastic loss of CPT1A. This is a pretty Western, but there are more variable ones also. So it's certainly, there's a band here. It's not 100%. And if you looked at the staining earlier of the mitochondrial trifunctional protein, you'll notice that it was in several different layers of the retina to some extent, but most strong in rod. And so this is kind of consistent with that. Okay, so we measured metabolism in, of palmitate once more in retinas from these animals. And, we just dissected them again, put them in a dish. Tissue utilization of 13C palmitate basically drops to almost nothing. And production of things that are derived from that 13C palmitate is, almost zero. And so that all sort of together says that of the things in the retina that could use palmitate, rods seem to be the primary cellular type that are doing that. And so that's kind of a result that I like. And there's no real change in glucose uptake or lactate production. So we're not really sort of changing glycolysis downstream of that or upstream of that, depending on your view. And here we have some slightly more functional data. Sort of the story is a little bit more filled in. And so, there's the OCT data. If you quantify outer nuclear layer thickness, it is lower by 10 months of age. But if you look at just the ICRE alone, at that same age, and this is John comparing it to his data, his published data, then you can see basically it's not really any different than the Cree model without any other transgenes. So, the Cree model does have a slight reduction in outer nuclear layer thickness over time, and that's probably accounting for the decrease in this model. But even with all that, it is a very minor sort of decrease. And if you look at ERGs, these are dark adapted, and you increase, basically the curves overlap for the controls and the CPT1A knockouts, the raw CPT1A knockouts. This was at 7 months of age. And so overall, I think the data are strongly suggesting that you can take CPT1A out of rods and you remove the ability of the retina to oxidize most fatty acids, or most of the ability of the retina to oxidize long chain fatty acids. And it doesn't really have a functional consequence on the ERGs, and it doesn't really have a functional consequence on the outer nuclear layer thickness.
Hey, Dan.
Yeah.
On your previous slide, was that just retina, or did you do iCup metabolomics too?
This is just retina.
Have you looked at iCups yet to see if
corresponding to the delta CPT1A in rods?
Yeah.
No, I haven't done that. What are you thinking?
I mean, we see like in the TSC1 model an influence or a change in RPE metabolism when we alter retina metabolism. Like they're just literally changing their basal metabolism as a result of rods changing their metabolism. I'm just wondering if they're doing something to compensate for the loss of this in rods or something.
Is that in like
Eric, you should clarify that it's the TSC is knocked out just in the photoreceptors.
Yeah, just in rods only. And
But
then
the RPE,
there's a
rod
effect in
has
RPE.
Effect, yeah.
And that's an intrinsic change at the level of like protein expression, or it's some kind of in vivo experiment?
You mean for the we did the TSC1 knockout in rods, which activates mTOR signaling, and then we take iCups and just did either targeted or 13C metabolomics, and you can see like a decrease in, I think it's TCA metabolites mostly, in the RPE. Like they have no genetic defect, but their metabolisms change dramatically because of a change in retina metabolism.
Yeah, so that's quite different than some of the data I've been acquiring in these AIPL1 knockouts that I often use as like a very severe retinal degeneration model, where we see steady state metabolites will change, and they'll change because they're not getting sort of fed by the retina, but flux of a labeled tracer doesn't change, because once you change the sort of supply side of things, then there's no real difference if you put them in a dish.
Yeah, I mean, this is all in vivo, like just pulling out and looking. We didn't do any ex vivo.
Okay, then that is very compatible with the kinds of things that I've been seeing as well. So no, we haven't done that in these mice. It could certainly be done. The focus so far for me has often been, how does altering glucose metabolism change sort of accumulation of metabolites in the RPE choroid corresponding to like a retinal knockout or something like that. But it's a worthwhile idea. It's worth testing for sure.
Dan?
Yeah.
This is Christian Grimm. You measured citrate that's coming from palmitate, basically here in this slide. So what is the percentage of citrate produced from palmitate compared to the whole amount of citrate you have? Is this a minor fraction now, what you're measuring, or what is the amount of citrate you have in total? And if you show a strong reduction, of course, but is this in relation to the full?
Yeah, it's a relatively minor fraction. So this is something where I guess I did not describe it very well, but if you take labeled glucose and you put a retina in that, you label the vast majority of metabolites, even with unlabeled palmitate there. And that just shows you that most metabolites are labeled from glucose. Palmitate can fuel metabolism, but it only fuels, a small proportion of it in this preparation. Mm-hmm. So this change is on the background of the retina doesn't use like an insane amount of palmitate to fuel metabolism.
Right. Thank you.
But just because it's not a major proportion of the citrate doesn't necessarily mean it's unimportant. So I didn't want to, like give that impression, I guess.
No, I completely agree, but I just was wondering what are you talking about? Is this in single-digit percentage range, which you now would use?
I think single to early double-digit percentage, if I'm remembering correctly.
Okay. Mm.
Hey, Dan, I have a quick question for you.
Yeah, of course.
So this is really interesting. So what you're showing here is with this CPT1A knockout in the rods that you have a considerable amount of palmitate that's not being broken down. So have you measured total palmitate in your samples? So where my question is going is, so if it's not breaking down palmitate, what are these cells doing with the palmitate that you're adding?
Yeah, I mean, that's kind of what I was trying to suggest in that Keen and the other author whose name I couldn't pronounce paper is that, here we go, is that, I think esterification to make fat esters is probably a much more prevalent pathway in the retina as a sort of output of palmitate, which is not to say oxidation doesn't happen. It certainly can. But, some is going to stay as palmitate. That's this 12.5. More is going to become fat esters. That's just 37.3. And then a proportion is going to be oxidized. That's just 10.2. So I think, they're all happening simultaneously, but I think perhaps more of the palmitate that's used is being used to make fats.
Has someone done lipidomics on the retinas of these mice to prove that?
No.
Because I would
I
think
don't know
if
if
it's like
lipidomics is the proof. I think more of those 14C experiments would show the extent to which there's a shift in palmitate oxidation versus esterification, because it kind of you can with 14C you can do like % of the dose that you administer, and so you can really see where that goes in a, I think, an easier way. Because with lipidomics, you'll see changes in lipids, but you might be missing, some pathways, and so you won't be able to sort of add them up and get a sense of what the total utilization of palmitate is.
Just to echo that, Mike, we are doing C13 tracing of palmitate into where else it goes besides beta oxidation, because we think the same thing as what's going on with that Dan and Jim and Nancy are reporting. And it's definitely, it's not surprising. It's well incorporated into lots of the things that you'd expect, phospholipids, tags, et cetera. But the quantification, how much is going into that versus beta oxidation, is really not that addressable by, a C13 tracing method for the reasons Dan laid out. And the old school radioactivity conversion into CO2 or H2O is probably really your best bet. So that's what we're trying to resurrect that right now.
Oh, I'm excited for that. Okay. So, yeah. So I've just showed you a lot of data that's kind of like, I would consider it borderline because I think it needs a little bit of work, but the overall message that, that I think we're approaching is that deletion of HMG-CSF2 or CPT1A, is not enough to cause degeneration alone, which means that, there are certainly physiological pathways, but the physiological pathway is not the only thing that's needed to sustain retinal health. It's not necessary, or I guess it's not sufficient for retinal health, it alone. And deletion of CPT1A from rods, again, you impair fatty acid flux, but there's no significant degeneration. And we have learned that rods in the retina seem to be the main site of at least palmitate utilization. And that in RPE, there are other cells that are contributing to that, that experimental output. And so what are we doing next? I think, all the things that you had suggested, but most importantly, just be thorough, have kind of up to at least a year, have all this data, including both ERGs and OCTs, and just make sure that it's all very solid before we can kind of make a strong conclusion, which I think we will be able to make it, but I can't say for sure at this point. And yeah, I mean, when the retina, so when if the RPE doesn't need to oxidize the fatty acids to maintain retina health, what happens to them instead? Are they exported, to the retina to make new outer segments, and fatty acids just kind of circulate between outer segments, and I'm talking about long chain fatty acids, not just the VLC PUFAs. Is there sort of circulation of the long chains as well? And or are some of them exported into circulation? And so I think that's just kind of a broad question of what is the ecology of these long chain fatty acids. And that's it. This is a non-comprehensive list of the people who have contributed to the to the talk, by generating a lot of the data and by talking about the data with me. And so yeah, and I'll take any questions that you haven't already asked.
I have an important question. Is that a flak jacket on Nancy?
That is, I'll let you and Nancy discuss that jacket.
It's an ice vest to increase metabolism. It really hasn't worked that well, so I wouldn't buy it. And I just want to say that this work with on the HMGCS2, because I think it was an oversight, making the HMGCS2 and CPT1A models was initiated by Kathy Bose Battaglia and myself, and like some years back. And so she has certainly contributed a lot to this project. And John worked on it before he left to go to Michigan.
Okay, I can read some questions off from the chat. So Monica is asking, I'm wondering why, if you know why HMGCS2 is expressed in the RPE cell membrane. Is it expressed? Oh yeah. So why the pattern of expression here is very, kind of almost membrane bound, right? Like that was quite interesting to me as well. I mean, I think a lot of mitochondria localized there. If you look closely, it's not just there. But I've not been able to sort of explain that result and why the localization is what it is.
The question was whether or not, to John and Nancy, whether this was bleached or not bleached. Because if it's not bleached,
it wasn't bleached. And this wasn't taken on like a confocal or anything. It was just on our on an episcope.
Okay. So there's lots of staining looks membrane-like when in highly pigmented RP, because the density of the melanin is just blocks fluorescence more centrally.
Okay, yeah, and I'm seeing that you had that comment after that. Okay. Rob Lensmeyer had that question too. Okay, for measuring metabolites, I indicated nanomoles per microgram protein. How did I measure both metabolite and protein in the same sample? You can, when we extract metabolites, we do so in a methanol solution, and there's a protein pellet that we essentially spin down from that and then separately do a BCA assay. And Larissa has answered that. Okay, so I don't even need to say anything in the chat, do I? Had some variability in recovering protein from the pellet. Yeah. Okay, so I'm now ready to answer actual questions. Let's start with Ronnie, who's at the top of my screen.
Yeah, hi, Dan. So I was wondering, you're showing us all these different models, and then when you're looking at retinal degeneration, you're not seeing any sort of evidence of that at the gross morphology level.
Yeah.
And so I was wondering, that could be possibly because the tissue or the cells are getting that source or that metabolite from a different pathway, a different source. And just like Christian was saying that, citrate can be produced from other pathways not necessarily involving the palmitate that you're feeding to the cells. So rather than looking at retinal degeneration, can you test for other pathways that might be taxed because you're deleting these genes? In other words, do they need to sort of upregulate some other pathway to maintain sort of homeostatic levels of these metabolites?
Yeah, I think, I mean, I specifically chose things that are kind of in a combined pathway. There are intermediates that will not be made because the only way to make them, I think, should be, well, okay. I can theoretically take, let's say for this RPE delta HMG-CS2, I can theoretically, if the ketones that are made from that are used to feed the retina, and I put the retina in the dish, it doesn't really test that anymore. I would be testing, you know, how well it eats what I feed it. And so I could provide, you know, labeled glucose versus, say, labeled beta hydroxybutyrate, but you know, it might start eating the labeled beta hydroxybutyrate at a sort of a normal rate because it doesn't necessarily react to the absence of the beta hydroxybutyrate, if that makes any sense. So I think the way to do it would be to like infuse the metabolite, probably. It's, you know, it's doable. It's just hard.
Yeah, because I was thinking, you know, with specifically respect to the ketone bodies, it's like you mentioned, right? You know, the RPE is sort of producing it and sending it back to the retina as a fuel source. But you know, if there's glucose enough there for the photoreceptors to perform, they might not need that extra source of the ketone body. It's only, you know, as Kira I think mentioned earlier, that you might need some sort of stressor for them to really show an effect.
Yeah, 100% agreed.
Yeah. Okay, thank you.
Okay, U004656.
Yes, I'm Miriam Baas from Leuven, Belgium. Very unexpected data, I would say. For the CPT1A knockout, I was wondering whether you can explain by the fact that peroxisomes can also oxidize C18's long-chain fatty acids, not to completion, but then it goes to mitochondria, but you don't need CPT1A for the short chain. So,
yeah, I mean, that's kind of, so we do show impaired oxidation in the 13C labeling. So while peroxisomes, I think, are capable of it, their utilization of that pathway of oxidizing, I guess you're talking about C18, not C16. Although, and so I don't have an answer to that other than it's certainly possible they can use an alternate pathway to oxidize C18, and it's something we could certainly test. But you know, we'd have to add it to this list.
And do you see any lipid droplets, or you still have to do the histology?
Yeah, I think lipid droplets are one of the next steps.
Thanks.
Roxana?
Oh, hi, Dan. This was amazing. Sorry, I missed the first part due to a conflict, but kind of going off what Ronnie and the previous person asked you about lipids. One, so you don't have it stress, but you do say that there is clearly impair of this fatty acid oxidation. Is it possible that you expect to have sort of accumulation of those fatty acids or other lipids that in time or with stress will probably be deleterious and you will see some cell damage, which you're not seeing at this time points? Do you expect that? Or have you thought to stress the system, high glucose, high fat diet, or something that, you know, kind of reveal this, you know, cell?
Yeah, so have I thought to stress it? Yes, that kind of came up before. One stress, especially for the ketone model, would be starvation. High-fat diet is also a stress we've discussed. I do not think that there will necessarily be lipid droplet accumulation. I think we need to test it to be certain. But you know, the RPE still has, you know, it still produces a lot of ApoE, for example, and it might be that it's capable of exporting lipids. Essentially, it interacts with circulation, and that might just be a sort of infinite source of drainage for excess fatty acids that are not oxidized. Or it sends it back to the retina. I mean, again, we don't really know the ecology of long-chain fatty acids.
Okay, I see. Any role of Mueller cell glia in this sort of
Well,
processing?
I would have told you that Mueller cells certainly must be oxidizing a lot of fatty acids, and then I got this result that showed that you know, there's very, very, very weak labeling in the absence of rod CPT1A. So I would assume that the Mueller cell kind of lives in the margins of being able to do this, perhaps just because it's not as abundant a cell type as the rods.
Yeah, thank you.
I got some people saying they got to leave. Steve has an announcement. Oh, yeah. I thought maybe that got announced before. But congratulations, Sri. Okay, any other questions? Oh, anyone else?
Dan, I'll just say very briefly to Miriam Bay's question of if you knock out CPT1A, is part of it that it compensates just by, you know, these sort of fatty acids that are in the 16 to 18 chain, of course, are not supposed to go to the peroxisome much, but under circumstances in many cell types, it's been shown to go there. And when we put on etomoxir onto our cultures with palmitate, we abrogate the initial spike in OCAR, but then the OCAR still is higher, and we're at doses that you and we have proven are pretty fully inhibiting, should be inhibiting mitochondrial beta oxidation. So we're now pursuing indeed whether there's a shunt when you're knocking out mitochondrial beta oxidation, whether there's a shunt into the peroxisome, which we know, of course, the RPE heavily relies on. You know, all the peroxisomal disorders cause a significant RPE degeneration. So that, I think that's a valid hypothesis for compensation.
But Jason, doesn't etomoxir also inhibit carnitine acyl transferases in the peroxisome?
It will not inhibit you don't need carnitine to get fatty acids into the peroxisome initially. So
don't you need it to get them out?
Mike can comment on this, and I don't want to hold up the whole conversation. We can talk about it later. My understanding is that they should have carnitine on to get out, but they don't necessarily have to. Mike's going to know more about that, but that's something I should also know. And before commenting, you know, I'm commenting without full knowledge right now.
Oh, Jason, just a quick question, follow up with your comment. So what is the contribution of the peroxisome FAO for the use of oxygen, comparing to mitochondrial oxygen use?
Yeah,
yeah.
We're super interested in that question because Miriam Bay was the one who just put out with their excellent grad student side, put out the PNAS paper that showed when you knock out peroxisomal beta oxidation, the MFP2 knockout, that was her paper, that you get this incredibly fast RPE degeneration, you know, very clearly proving how important peroxisomal beta oxidation is. And if she had a cool sort of experiment where she created a RET degen model that's very fast so that there was no longer any phagocytosis happening. And when she crossed it to that RET degen model where there wasn't any phagocytosis, the RPE defect was rescued, strongly suggesting that it's the handling of the incoming outer segments, which of course have all the VLC PUFAs and DHA, that's critical for inducing the toxicity when you don't have peroxisomal beta oxidation around. And they did even more to show that it was the VLC PUFAs in particular that were impacting lysosomal capacity. Anyhow, the bottom line is because of that work, we're really interested in the crosstalk between the two, but the tools for peroxisomes are so not great compared to the mitochondria. So it's a lot more work. It's not like sprinkle on a drug. It's a lot more work. But anyhow, I think that's something to keep in mind when we're thinking about like what happens when you knock out mitochondrial beta oxidation. Is there some compensation with the peroxisomes which are robust in the RPE?
I think that's where the labeling comes in, right? I mean, we could do that labeling with stearate and really see the extent to which it is lost versus if there's compensation, we'll also see that.
I agree.
All right. Thanks, everyone.
Thank you. Bye-bye.
Thank
Thanks, Dan.
you.